The idea here is that we understand aging fairly well in humans, and we understand the kinds of cellular and morphological things that change with age as we grow older. And what we try to do is understand how in nature, in natural species, things might happen slower. So we understand in humans about cellular deterioration, or say muscle decline or joint problems. And there are a lot of species in the natural world that either have those same processes, but they happen a lot later in the lifespan. And in particular, I study reptiles. I'm mostly looking at turtles and snakes because we've identified populations that vary in their aging rates and vary in the cellular processes that contribute to sort of old age mortality and setting lifespan. And then in particular, getting going and getting started here, I'll be initiating projects and research on sex differences in aging. So why do, in some species like humans, why do males live shorter and age faster? In other species, it's actually the females that age faster and live shorter. And so we're very interested in how sex or gender affects aging rates and biology of aging. So the idea here would be that if you want to understand aging biology and intervene in humans, one way to do that is to study specific mutations in sort of model species like mice or lab rats, where you can manipulate a particular genetic pathway. A complementary approach is to look at those same pathways and how they're functioning in animals that haven't been manipulated. And in particular, reptiles have, on average, slower aging processes than we see in humans and things like lab mice and lab rats. And so they make a nice contrast. And we can take what we learn, for example, looking at cell repair, and say, how does it happen in painted turtles? How is it different than how it works in humans? Or is it the same but just happening slower, and why and how? And can we use that then as a targeted area of intervention for humans?